Hello, everybody, and welcome to today's Mountain Talk. I'm Jennifer Cunningham. I'm the AVP of Alumni Relations here at Lehigh, and I'm here with Dr. Xiaochen Gua, who is an assistant professor in the Department of Electrical and Computer Engineering. I'll tell you a little bit more about her in a second, but I do want to go over the choreography of today's Mountain Talk. We're going to go for about 30 minutes, and the first 5 to 10 minutes, Dr. Gua will go over her slides and tell you a little bit about her amazing research that she's working on, and then we'll have time for your questions. The way that we collect your questions is through a chat box. So if you take your mouse and you scroll over the screen, you'll see a bunch of icons pop up on the bottom of your screen, and one of those icons says chat. It looks like a little cartoon bubble with 3 dots in it. So click that and you'll see a window pop up that says Zoom webinar chat. So you'll just type your message where it says type message here and hit enter, and I will see those questions and I will read those aloud. So before we get going, I'll tell you a little bit more about Dr. Gua. She got her PhD in electrical and computer engineering from the University of Rochester. So she's very used to these 10-degree temperatures that we're having here today. And she received her BS degree from Beihang University in China in 2009. She's going to talk to us today about some work that she's doing on a newly funded National Science Foundation project focused on engineering a neural network. The goal of this project is to use optogenetic in vitro neural networks to run learning applications. She is an NSF Career Award winner and has won the IBM PhD Fellowship 2 times. She's won several other awards that you can read about in the bio we sent out about her before the webinar. So let's get going. Dr. Gua, go ahead and get us started on the slides.
Okay. Hello, everyone. Glad to be here to talk about our recent research on the living neuron machine. This is a collaborative work with our students, Yuan Zeng, Zhu Beiyer, and 2 other faculty members at Lehigh, Evgeny Berdichevsky, and Zhiyuan Yan. So recently, computer architect has been looking at mimic the biological neural networks. Why? Because biological neural networks are capable of learning and they are also power efficient. So we simplified the neural network to build a neural network in silicon. Computer architects are building different neural networks such as feed-forward neural networks, convolutional neural networks, as well as recurrent neural networks and all other neural networks to solve complicated learning and prediction workloads. They are useful but not energy efficient because they are compute intensive and memory intensive. And also they are very different from biological neurons because they are, first of all, simplified. Second, they have some topology that are different. And also more important, they also have different properties like learning and predictions. So we have been thinking about this problem of how can we actually better harness the power of something like the brain or neural network or living neural network to perform learning tests, to compute learning tests to be energy efficient. So we look at what we have, and we fortunately found a collaborator that have a wet lab that can actually manipulate living neurons. And then we thought, maybe we can build a machine out of living neurons. So we call it a living neuron machine. So how do we build it? So we have a dream, right? So this is a picture from Star Trek, and this is the Bell neuron neural pack. So sort of this is where our idea actually get inspired from. So in this Bell neuron neural pack, you can see there's neuron fibers. They connect to, they live in a gel because they need nutrition. They connect to metallic interface. We want to build something like that, but we have to start with something simple. So our machine looks like this. So we grow some neurons in a dish. We dissect those neurons from rats, and they actually grow themselves, given the right temperature, the right nutritions. They connect random networks. And we can use some chemical to label them. We can also put some barrels in the dish so that they are optogenetically responded to different wavelengths of the light. So this way we have optical input. We can also mark the cells to be able to detect the concentration, because when neuron fires, the concentration changes. So this way we can also detect the neuron activities. And to detect it, we also use optical interface. So we have optical interface input and output in this experiment. So this is our next slide, shows our experimental setup. The cells are optogenetically infected, so they are labeled individual cells, and they can have optically controlled ion channels, so that we can stimulate selected cells. And we can also program those cells by using a technique biologically proven called STDP, by timing dependent plasticity, to give them different connections. Of course, if there's no connections, we won't be able to force the connectivity. However, if the connections are already there, we can increase the strength or decrease the strength. And finally, we can have optical detection, and we can also put them into pattern dishes, so that from engineering perspective, this gives us modularized design so that we can look at a less complex system. So in this system on the right, the stimuli is given through a light pattern to this microscopic objective, and it is going to stimulate some neurons. In this first attempt, we are going to only record the first level response from the neuron, so that we know that there's no complication in terms of recursive response. And then we detect through shedding a different light to detect the causing concentration label. And then the responding fluorescent light will tell us which neuron has fired. To give you an idea of what's the potential application in this experiment, this is a simple illustration. So we can do a very simple artificial intelligence task called handwritten digit recognition. So there are different handwritten digits, different zeros, different ones. We are supposed to recognize through image processing which images are zeros, ones versus twos or threes. So first, what we do is we take a picture take an image of one of the handwritten digits and translate different pixels to spike trains. In this experiment, we just interpret let's say, white pixel into there's one spike, and then black pixel into no spike, so that we can stimulate predefined input neurons. And then we can detect the response through the predefined output neurons. We can observe their activities. And if the prediction is incorrect, we will train it according to back propagation or stochastic gradient design algorithm to strengthen or weaken the connection. So this is our proposed approach. And currently the research is still undergoing. We have learned a lot about neuron characterization and algorithm design, but we are still waiting for the neuron to grow in proper conditions so that we can actually test our algorithm on the wet lab experiment. That's what I want to cover today. Let me, if there's any questions, please ask.
Yes, so participants, again, you can type a question by going to the chat box at the bottom of your screen and type your message there. While you are thinking about your questions, I have some. So tell me a little bit about how you conceived, like how you thought to do this. Was this something that you had been working on some research prior to this and it was an outgrowth of that, or did you wake up one night and say, aha, this is, or were you watching, was it the Next Generation and the Borg, or
right?
The Star Trek?
So to give you a little bit about my background, I'm a computer architect. So normally I build computers using solid state silicon, sometimes emerging memory memory technique like resistive memory. There are a lot of research activity in my domain about building neuromorphic computers. They a lot of research ongoing, working on building memristor-based neural network to mimic the neural network behavior to compute deep learning applications. Well, there are a lot of value behind that. They are already achieving better energy efficiency as compared to general purpose processor. But we want to go a step further. So we want to actually understand how neuron does certain things and whether what people imagine they are doing or they are capable of doing actually is what they are doing. So one day I talked to my colleague who is working on a similar area about this question, and he actually doesn't know. So but he is very interested in figuring it out together. So that's how we established this collaboration and actually work on this project.
Okay, good. We have a question from Brian Force. What types of problems, for example, image recognition, classification, are most suitable for biological machine learning? And what applications look the most promising for this type of solution? So
right now we are focusing on this handwritten digit recognition. The reason being that the handwritten digit recognition actually is inspired from the cortex neurons. So I think in the long run, so right now I don't have the exact answer, but in long run, I think it depends on the type of neuron, because there are actually different constructions, different topology for different region of the neuron. So the cortex neuron will be a little bit different than cerebellar neuron. So we are also thinking about being able to use different cells, but there's also a technical challenge. So that answer depends on which region of the cell we actually use.
Right, okay. Joe Bailey's question, do you expect faster results using biological systems? Is training going to be faster? Will training results from biological systems be transferable to digital systems?
That's a great question. So there are a lot of debate in the AI community about what the neuron is actually doing in terms of training. Because right now the neural network is doing backpropagation, and multiple researchers is thinking that the actual neural network is not doing backpropagation. And we sort of believe similarly, but right now, given the constraint of our experimental system, we won't be able to figure out that question yet. So right now we are restricted to single layer neurons, and we hope we can answer that question in the maybe next project or later when we finish our single layer neuron experiment.
Yeah. How long do you think it'll be till you finish?
So right now our goal is to finish it in 3 or 4 years.
Nice. Yeah. John Doherty asks, biological processes can certainly be reliable and standardized, but biological cells inherently have the ability to behave independently, thereby introducing inaccuracies. Is this a potential issue? If so, how can it be controlled for?
Right, so this is definitely a challenge we are facing right now. Right now our solution is replications, and which include both spatial and temporal. So right now our experimental team is doing the calculation, and we are sampling different cells, and we are also sampling each cells multiple times to give a statistic estimation about the distribution. Our computational team are right now using this information to understand how much inaccuracy this is actually bring. So in the algorithm perspective, the traditional artificial neural network, or the network that is doing the recognition, has redundancy built in them already. So we hope that by controlling the statistic variation and understanding the statistic variation and controlling the degree of duplication can help us to overcome this particular challenge.
Okay. What other big challenges do you have Are there a couple that if you could solve those things, you'd be set?
Right now, it's really about encountering some challenges and overcome them, and then the next couple will come.
Yeah.
So right now, actually, our challenge is experimental setup. So the experimental team have been spending a lot of time to figure out the right virus to use for the optogenetic cells to infect them with the right virus so that they can respond to the light. A particular virus we used in the past will infect both the soma and dendrite, which will cause a lot of inaccuracy when we try to simulate individual cell. Right now, we've changed a different virus so that it only infects the soma. So only the soma is responding to the external stimulation, and that helped a lot. On the computational side, we are also right now in an initial exploration phase. So right now, we tried a couple of algorithms by using this bioaccurate simulation, but the accuracy is not actually as good as the computational system, because right now we are trying to adapt a artificial neural network or computing based computation driven model to this biological model. So we still have to figure out the best way to actually develop the algorithm for the biological neural network.
Okay. So John also added, are there opportunities to get involved with this research? He's very interested.
I think so. Certainly send me an email. We will figure out some
how alumni can help. Yeah. I was wondering about the viruses that you're looking at and injecting. Where do you get those viruses? Is that a partnership with a different university? Is it here at Lehigh? How do you decide?
I think my colleague purchases them from a company that's working on it. And I mean, some of those biotech companies, they don't have a long lifespan, so they emerge and they disappear, they get acquired. So that's also a challenge, how to secure a particular source of this kind of resource.
It has high quality and all that.
Right.
Yeah. So other universities, is there anybody else working on this?
Right now, we are the pioneer on this. Other people think this is too risky. I've talked about this project to a couple of other faculty members I've known in my community. They think this is very risky and high reward as well. I think there might be a couple faculty, maybe there is a group in Georgia Tech that is working on more on the biology side, not on the learning side. And in the past, I think there was some paper published on using this patent biological neural network to do some recognition. What they did is to build this electrode array to attract the soma to those different electrodes so that they can make sure that they know the topology. So right now, our network, we want this neural network to be able to freely connect to among themselves so that it behaves more natural. Whereas in the past, people have tried like forcing them to connect to each other, either fully connected or connecting to a neighbor. They've accomplished something, but I think what we are doing we might discover something new.
Yeah. Peter Tedesco asks, let me ask you, a futurist, and perhaps do a little dreaming. Where do you see this technology bringing us in about 20 years? What kind of applications do you envision?
If I go really crazy, it would be that Star Trek bio-neuron germ. But to be realistic, I think one thing that definitely in my area, people will pick up on will be how to actually build a silicon-based replica. Because right now, we are just imagining how neuron works. And if our research is fruitful, discover something that the neuron is actually doing, and because it is already proven that the brain is more energy efficient. And right now in computer architecture, energy efficiency is the critical barrier, so to improve the performance of the computer. So anything we discover, I think, will be quickly adopted by my colleague in my area in computer architecture to build the next generation energy efficient deep learning accelerator, or just learning accelerator, or neural network accelerator. From the biology point of view, we think because we do make quite a few we will make quite a few contributions just to make sure that just to make this experiment possible. So we will publish the protocol that we actually use to make this possible. So this will give a test bed or experimental mechanism for the biology scientist to actually understand neural network at this particular scale. So right now, the cell level biology of the neural network is well understood, and maybe people understood like a couple of neurons put together what happens. There are a lot of hypotheses people try to prove, but has not been able to. And hopefully our contribution to the experimental side is going to help them to accomplish more.
Nice. So you mentioned that other people aren't doing this. They think it's too risky. What are the risks?
Being the first in the field is just nowhere, you know where you're going, right? So initially, we just thought this is cool, and we don't even know how many challenges we are facing. Like I mentioned earlier, we have to just solve one set of challenge, and then just another set of challenges are thrown onto us. So right now, the challenges that I think are considered risky is first, the network topology is unknown. I think there are theoretical people who have dealt with unknown topology and how to make use of it, but nobody had actually tried to do experiment on this unknown topology in wet lab experiment. Being able to, or if we have to work on something that maybe there are certain fundamental things that hasn't been understood, that is also high risk, right? So for example, when people work on any solid state machine, they've already known how transistor behave, at least at the scale the details they wish to know, for quantum computing, people probably also know something about the physics. And for biological network, we know the cell, how it works, and there are theories that are mature. But if I look at the publications on network studies, there are hypotheses, there are different ways people publish papers to prove this hypothesis. So, but we don't know for sure because there are so many variables. There are different type of cell, there are different type of environment, there are different type of equipment. Basically, there are just many things we have to figure out ourselves. It's not like there is a giant that we can stand on top his or her shoulder. Right. So that's why it's risky.
So to boldly go where no one has gone.
Right.
Thank you for taking that leap. Joe Bailey asks, will this research help us with our understanding of how the brain works?
In part, right? So we are not looking at the brain scale neural network. So our experimental setup is about 100 to 1,000 neurons. So how brain works is another topics, and different people are looking at different scales. There are biologists that look at the function areas, functional district area of the brain, how they communicate each other, that turn out to be quite complicated. They use other tools to study the stimulation at larger scale. And there are other biologists that looks like the individual neuron. In fact, that's also well studied. And also there are people that looks like similar scale, but not really looking at the learning aspect. So I think we make a tiny contribution to that aspect, but I don't claim that we are working on the entire problem.
Lori Natoris asks, what programming languages and platforms are you using in your research?
Okay, so that's right now we are using a simulator called Neuron, which is a functional programming language that is tailored to neuroscientists. And we also combine that with Python and other tools. So basically whatever the students feel comfortable with, we allow them to just use those tools, right?
So you mentioned students.
Right.
You're primarily working with graduate students?
Right. So we work with both graduate student and undergraduate student. Last year we had 2 undergraduate students working with us. One from CMU, one from Lehigh, and one was a visitor from China. So this semester we are fortunate to have another Lehigh undergrad joining our team, and also another visitor international student. As an undergrad student. And right now we allow our graduate researcher team a little bit larger. So we had 2 last year, and this year we have 3.
Very nice. Very nice. And what do the students do once they graduate? Do they work in this field, or do they continue researching the same thing? What do they do?
I mean, it's up to them. So right now, this is a collaborative team. Our students also have very different backgrounds. One student is primarily electrical engineer. Another is computer engineer. And then the third one is bioengineer. So they have their expertise in different domains. I mean, many of our Lehigh graduate students, after they graduate, they go to companies. Some of them find academia jobs. Some of them may thinking about going to find an academia job, but they choose to first do a postdoc. Some of them may be doing maybe go to a industry research lab. But essentially, I think the training they will receive by being part of this project will be useful regardless of whether they work on the same project or similar project or not.
Yeah, neat. All right, well, our time is just about up. Is there anything we didn't talk about or questions people didn't ask that you would like to talk about? Anything alumni out there should know about your work?
I just want to mention that this is really a initial step that we are taking. So there's not too much result yet, but we hopefully we will pick up the pace quickly. But we really recently just have 1 publications, and we don't have a lot of answer to many of the questions you have. And thank you for supporting us and watching this video.
Yeah, well, and thank you for sharing. I know it's sometimes risky to share your initial work when you don't have a lot of, things like results yet. So that's very brave, and we're really glad that you took the time today to come down and share this with us. So thank you, and thanks to everybody who participated. And we look forward to seeing you on our next Mountain Talk.
Okay.
Bye.
Bye.